The genome is very much like a book. To actually read the book, you tear it up and you read all of these fragments that you've shredded the book into, and then you have to piece them all together to reconstruct and reassemble the book.
In 1997, in a moment of serendipity, two then unknown chemists laid the groundwork for a genomics revolution. While conversing over a pint of beer at a local pub, they stumbled upon a groundbreaking idea that would dramatically reduce DNA sequencing time and costs.
When we came up with the idea, we had very little or almost no data to show that it would work. So we were left with this quandary. We had a very good idea, very exciting, but no data to support it.
David Klenerman and Shankar Balasubramanian had only recently got to know each other. After completing a postdoc in physical chemistry at Stanford University, David had returned to the United Kingdom to work at BP Research.
The idea was to take cutting-edge scientific methods and apply them to problems in the oil industry. And I had literally a lab with a large amount of equipment and a lot of freedom to do what I wanted to do. I think I expected to just stay on at BP as a career, but that didn't happen because there was a big reduction in the oil price. So eventually, BP moved me and the equipment to Cambridge University. I was looking around what to do.
Around the same time, Shankar had just completed his successful postdoc at the University of Pennsylvania. He intended to remain there, but Sir Alan Roy Furst, a chemist based in Cambridge, convinced him to change his plans.
In a sense, he was saying, don't be too persuaded by these Americans. You can do world-leading science here. I thought, how can I not listen to what he's telling me? So I made the decision to come back here and give it a try.
Shankar returned to Cambridge, the home of DNA pioneers like Watson and Crick.
DNA is always in the background in everyone's mind. In this place, you see it everywhere. The project itself came about as a pure accident. I wasn't crystal clear on what I would work on. So we had a lot of discussions about what could be the next big transformation in the field. I tried a number of different directions.
One of the projects involved studying the cellular genetic copy machine, DNA polymerase.
We had got some results that were nice. We wrote a paper and we submitted it, and a referee asked for a time-resolved fluorescence measurement as a control. Now, ultimately, Chris Abel, my former supervisor, who was at Christ College, he said, you should talk to David Klenerman. He has all these lasers. Then we kept talking, and that's when we conceived of this experiment.
DNA is a code of 4 bases. We can color code them, and we just have to work out what the color is there. And the idea was use single molecule fluorescence to look at single molecules, which was very exciting.
To watch a polymerase extend DNA in real time. We wrote a grant. It was funded.
The experiment was, like most experiments, harder than you think, and you come across unanticipated problems. We had to watch in the right place at the right time to see this fluorescent signal and see it change. And one of the frustrations on our side is we kept on missing this. We were using a confocal microscope, which just meant that we were only looking at one point on the surface at one time.
Some of these discussions we had down the pub at the Panton Arms. Piece by piece, we started to see alternative ways of sequencing DNA. Instead of just observing single molecules and watching a polymerase in motion, if we can control the process of nucleotide incorporation by the polymerase.
Meanwhile, in 1997, researchers were still struggling to sequence the first human genome at the nearby Wellcome Trust Sanger Institute.
The Human Genome Project was an almighty project, very expensive and time-consuming and hard work to get one genome. We have a planet with billions of people. Everyone has a genome that is different from each other. Understanding the details of genetic variation on a population scale ultimately would need sequencing at scale. And there wasn't an obvious step from that to getting lots of genomes.
I started to wonder what happens if I had a camera that could look at the whole surface at the same time.
The true genius and simplicity of the idea behind David and Shankar's new sequencing method lay in its parallelization. Instead of sequencing a single molecule at a time, could it be possible to sequence millions simultaneously?
We use the reference genome to help guide us as to how to restructure and stitch back together all of these fragments. We put all this together and we came up with a figure that was somewhat surprising.
So I remember doing this calculation on a flight to Germany for a friend's wedding. And when I got back, I wrote it down on a sheet of paper and put it in Shankar's pigeonhole. And then within an hour, Shankar came down to my office.
We were starting to get quite excited. Here's something that we could do 1,000 or 10,000 times faster, better than the current methods. And it felt achievable.
I was attached to Christ College, and the master there, Alan Munro, had actually previously worked for a venture capital. He put us in touch with a venture capital firm, Abingworth, and they didn't know quite what to do.
I was very junior, as was David, and no track record, nobody knew who we were in the scientific world. Yet we had built this vision to create a system that could potentially transform genetics.
And eventually they decided to fund us at a very modest level to get started, to do some simple proof statements, to de-risk the project. And that's how the company started.
In 1998, David and Shankar founded the startup company Solexa.
The key thing to get it working more efficiently was amplifying the single molecule of DNA into a colony of 100 copies, and that just meant the signal to background was better. We literally went from an idea done on a piece of paper in 1997 to having a fully functional instrument 10 years later. Me and Shankar were lucky enough to initiate the whole process, but it's really underwater technology that wouldn't be possible without this large team of talented people who took an idea on a piece of paper and brought it to reality.
In November 2006, United States-based Illumina offered $650 million to buy Solexa, making the project one of the greatest commercialization success stories to emerge from the University of Cambridge. And in November 2008, the sequencing of the first African genome was published in Nature, alongside the first Asian genome and the first cancer genome. Today's
sequencers do multiple trillions of bases per experiment. So we're now over 1 million-fold improved. And I think in experimental science, million-fold enhancements don't happen very often.
We did think about how people might want to use it, but the community used it in many more inventive ways than we imagined. And so the impact, it's gone well beyond what we expected.
The monumental impact of next-generation sequencing reaches beyond the lab, driving progress in medicine, agriculture, forensic science, and the battle against infectious diseases.
The best-known example of infectious diseases is when the SARS-CoV-2 virus was sequenced using next-generation sequencing, and allowed over 1 million cases to be characterized in terms of the sequence of the virus and the changes to the sequence. And this has informed mRNA vaccine design in real time.
I saw a program on the BBC. It showed a young child being born and was unwell. And what they'd done is sequence the mother, father, and the young child. And then they looked for mutations in genes that might explain why they had this disease. They could work out what was wrong with the child. There was a treatment and the child could go home. And it brought home how impactful the technology was. It's not just a way of sequencing DNA, it's literally a way of saving someone's life.